Prior to our access to the AmpliSeq panel, it was really difficult to measure a large number of DNA methylation events in a single sample. It was difficult to get accuracy, reliability, and high-quality data. So with the AmpliSeq panel, we get single base pair resolution, we get percent methylation, and we get really precise measurements with much tighter error bars than what we were getting with methylation-specific PCR, the competing technology. We had spent a long time trying to get methylation-specific PCR to work, and we found that we were needing to set up hundreds of thousands of PCR reactions to do the kinds of studies we wanted to do. And this was not only a problem in terms of how much work it required, but it was very difficult to get that many samples processed properly and compare them all across the different batches required to process them. So we're getting something like 100 times more information out of a single tube than we got out of hundreds of tubes using methylation-specific PCR. Methylation is a relatively unexplored area in cancer research. Preliminary data is showing that it's really good at telling you all sorts of things, including what lineage the cell came from. So it can be used to tell the difference between breast cancer and colon cancer, for example, in circulating tumor DNA. That's one really important application that I think would be great for this kind of panel. Often in pathology, we might have two different kinds of brain tumors that need two different kinds of treatment, and they differentiate along different cellular lines. Methylation should be perfect for picking that up. Until now, there wasn't a good, reliable assay that could be done, let's say, in a small panel of genes where you know what you want to look at and get really good high-quality data. Essentially, there was no way to get really good high-quality data that was reproducible and usable by your average user.